Hello everybody, welcome to the second class of the cell biology, molecular cell biology course. I hope you still have a great time, uh, yeah, and in health and your spirit also. I just remind you one thing, because maybe a lot you will be considered for the midterm and also the final. You need to follow, you need to read the book. Like I mentioned last time for guiding, because you need to the easiest way to digest the book is you need to read, try to read each chapter, but not read any text in there. You need to just dissect and read the title of the section, and then try to look at the figure of the section and try to identify what the meaning of the figure. The picture is very easy to grab. And if you don't understand some part, you just need to look at the text or discuss with your friend or ask me in the blackboard. Post your question there. I hope I can answer it for you for that kind of thing. Okay. In this today topic will be characteristics of the cells, classification, and I will fly through the organelles of the cells. So this is you already know, I think, but I just remind you some part before we can go deeper some part of the cells. And in some slide, I will explain a little bit about some chapter in the book. We will focus on more the deeper. Okay. So he is I list a search on a book and also another book and looking for you some basic properties of the cells. Here you can see this. The first cell is a complex system of the universe, and the one of the most important property of the cells is the living. They differ from something dead. So, for example, something is we cannot call it a living thing is a viruses. But you see, the viruses cannot living themselves independently. Without the host cells, the viruses will be die. Let's say some coronavirus can survive in your phone about 8 hours, but more than that they will die. But the real cells will be survive if they can find some environment, a suitable environment. And in viruses, a suitable environment is the host cells. They need to relies on another source of the living. And the next is cell is very highly complex and highly organized. You see in the structure of the cells, in the animal cells or the plant cells, very complex, right? For the outside, with the cell wall, cell membrane, with a lot of organelles in there, and the way they regulate it. Next is the cell has a genetic program and the means to use it. We're talking about DNA and a lot of tools to translate it and transcribe it to RNA and translate it to the protein with a lot of the protein molecules. So knowing the process, knowing all of the molecular protein and the function of that protein is one of the main topic of this course. And next one is cell is capable of producing more of themselves. So I mean the cell can provide themselves. I mean from one cell to become 2 cells, or 4 cells, 8 cells. They're doubling. The cell can do it. So without that process, the cell is only one cell. The single cell cannot provide themselves. Reproductive is no. So they cannot survive with the environment. They will die, right? And cell acquire and use the energy, of course, because cells is quite open a system in this universe. They have to follow the rule of energy in universe by follow the rules of energy. So energy is not from this point to go to another point, not just automatically created from nowhere, nothing. And cell carry out a variety of chemical reaction. Of course, you can see deeper in the further chapter. So a lot of reaction happen in the cell. You see it. When you culture the cells, you just see the simple cell membrane, something. But inside that cells, a lot of things happen in any minute. I mean, any second, very fast. Some reactions are very fast, some reactions very slow. And next is cells is have some things activity related with the mechanical activity. In the bacteria, you see that right, the bacteria can move it from the current position to another position with have a nutrition or something. They can move it using the plasma or something. How about the animal cell or the plant cell? They also have ability to move too. The plant cells, you will see the video, and in the animal cells, like you can check that using the picture, the video of the immune cells when they're seeking for the bacteria inside of your body. Okay. And next is cell able to respond to the stimuli. Yeah, in the bacteria, when you get some kind of heat, the bacteria have some kind of reaction, or if you got put some sugar or nutrition into the bacteria, they also have some response. And the same thing, but in higher complex level in the eukaryote, especially the animal cells. So it's a lot of signal come to cell any minute, and the cell need to be respond properly with that kind of stimuli. They need to balance with this signal and another signal, and that's why is our tissue kind of community of the cell become tissue. It can work perfectly. And next is cell have the capable of a cell regulation. Yes, for example, in the bacteria, if they are in the they're living into the environment without the less nutrition. So a lot of gene in the bacteria will be changed for expression. Maybe they will upregulate some gene, up expression some gene and down some gene. Make the bacteria will be maybe less active to save energy for for example, and the same thing for the eukaryote cells. And the cell can evolve, yeah, thanks to the DNA. So the DNA is you can see that for in our planet we have 5 billion years already. So from the very simple cell to become the very complex cell right now. So they have evolved, and that information is we know it is stored into the DNA. Then DNA now from very short from the beginning to become very long and complex and right now. But I don't know. Maybe we can find another molecule can store that information of the evolution process, and that's different with the DNA. We can check it. And cell can grow and reproductive in culture for a 10th period. This is how we say that. This is not true for all of cells, because a lot of cells we cannot culture it. But in the science, especially in the bacteria and in the biotechnology, we can culture the cell, we can culture a lot of type of cells, but not all of them. Okay, so we still have a lot of property different types of the property of the cell, but you can find your cell's property. But this is just some very basic one. And different cell type have a different property. They different. And that's kind of finished for the properties of the cells. And the next thing, we go to a little bit touch with the classification of the cells. I also go fast about this part because in biology or in high school you already know about this. So prokaryote. So pro is before and karyote is the nucleus. So you touch this one, like a technology like karyotype. That means you look inside of the nucleus to see the any chromosome inside of the eukaryote cells. So you see that bacteria or prokaryote are very diverse in the shape and the size also. The size is quite not much different, right? But the shape is very diverse and the function also very diverse. You can see it in here, the global shape. This is a rose shape and this is a spiral shape. And each have a very diverse localization on the Earth, and also very different in the function and different in the activity. So this is some common characteristics of the prokaryote. So prokaryotes are the most diverse and numerous cells on the Earth. You see that biotechnology brings us a lot of the application using the micro prokaryote for doing a lot of good stuff for the human. But right now, the scientists have to admit that a lot of bacteria we don't know any information. We just know just a little bit about the micro prokaryote type. We have maybe 90% we don't know about, and 10% I don't know exactly the percentage, but that some bacteria we know, but a lot we don't know, and a lot of them is we cannot capture it. That means that we cannot do any research about that. But the good thing is now a new technology can help you to check that. I explain later. For we can check the bunch of microbes in one position of the soil. For example, we can check the DNA of all the bacteria in that kind of soil, that kind of sample. So we can analyze it. If you're interested, we can discuss this part later. So the some as common characteristics of the prokaryote is small size, having cell wall, like I also quite want to tell about the cell wall of the bacteria. You see that if you look at the bacteria under microscope, you can see a microbe quite very small and look like the egg. They can easy to break, but not. The cell wall of bacteria quite hard. And whenever I try to produce the protein in a bacteria, normally I need to use some enzyme to eat the cell wall before I have to I can break the cells. Yeah. And of course, a bacteria have no nucleus membrane. They have nucleus, but they don't have any membrane in the cells. They have the DNA, they have nucleus, but the nucleus and the DNA floating freely inside of the cytosol. And compare with the prokaryote, so they have very fast division, about 20 minutes for the bacteria like the E. coli, Escherichia coli in the human gut. 20 minutes we have the doubling one time, very fast. But this sometimes is not true. Some kind of bacteria is very slow to grow, maybe a week. or a month to grow up. So fast division, very important characteristics for biotechnology, right? Because of a lot of scientists want to use this kind of system, bacterial system, to help us to produce a lot of type protein. And they have the diverse characteristics in the biology. Diverse characteristics in the biology. Yeah, different types of bacteria have the different characteristics. They use oxygen. Some is not use oxygen. Some use a very sweet. Some is use acidic chemical for food. And they adapt diverse environment in the earth. So from the very hot environment to the cold environment, from the surface of the earth or very deep in the seas, we can see the microbe in there. Even somebody see the picture of the microbe, evidence of microbe in the moon also. And they can use a diverse material as a food, yeah, because of different places have different food. China and Vietnam has a different food in there also. In here you can see this, the picture in the bottom, the left side, you can see this is using transmission electron microscope to see inside of the one E. coli. This is transmission electron microscope. You can see the picture like this inside of them, and we have to see that no any organelles in there, just a bunch of the material in there, like DNA or something. So the question is, do we have any kind of microbe prokaryote have the mitochondria? You know, mitochondria is very important to the cell, right? To make the energy for the cells. They make the energy for the cells. But why is the bacteria don't have the mitochondria? Without mitochondria, how bacteria can survive without the energy? So the thing is, the bacteria is actually they don't need the mitochondria, because somehow they use the membrane for their own membrane to make the ATP, and they can use the glycolysis. So if somebody still remember something related with the biochemistry chemical, so you can remember bacteria will induce the glycolysis. So you don't need the glycolysis also produce about 2 or 4, maybe 2 molecules of the ATP when you break the 1 molecules of the sugar. So let's say, so you don't need the mitochondria to produce the ATP. Or some bacteria can use a membrane to produce the ATP. Okay, next is prokaryotes is a fundamental part of the life. Yes, they're very important with the living thing in the earth. So I believe that we come from the ancestor, but microbe is really contribute to the development and evolution of the eukaryotes. I mean, it's all of the modern cells in the world right now. And also, they are also in the current time, they also contribute a lot to the nutrition and digestion and energy transfer for the life in the earth, because they very diverse. Like for example, some kind of cells, bacteria in deep sea, they can use a hydrosulfur, the very toxic gas, for their energy. So, and they go to the food chain of uh, the ecosystem. Like here, it's like a very important thing and also very basic. The plant can fix the no no the bacteria in the plant can fix the nitrogen, and that nitrogen turns to the nutrition for the plant. Um, Another and very important um, thing in the classification of the prokaryote is archaea. Archaea in here. So what's the archaea? So that is also prokaryote. Why? Because they don't have the membrane of nucleus. They don't have the nucleus membrane. So they form another group. Another group. They are kind of morphology. I mean the shape and something here. This is archaea. They seem similar with bacteria, but inside different. The scientists identify the genome is of the DNA of the archaea is quite very different with the bacteria. So normally archaea is kind of old, very old bacteria, and they live they live into very deep and very hostile environment, maybe very hot in deep sea, very very hot one. And what are characteristics and kind of different with bacteria that have intron? So remember in your genetic class, so that in bacteria we have the polycyclonic gene, right? With the one promoter, and one promoter can induce a lot of gene expressed, so they don't have intron. The virus also the same thing. Virus we have a polyprotein, so all the protein they synthesize under the control of one promoter, and after finish making the protein, some enzyme will come and cut that big protein become the small protein, different with the human have intron. And they also have a complex RNA polymerase, the same with human, same with the animal. Because of the very, very interesting characteristics, archaea is now quite important in the biotechnology. Because of the, you can see that with very hot environment, this is archaea can survive. So we can somebody knows about tag DNA polymerase, right? So we can use this one the same meaning. So we can isolate some enzyme from the archaea and use for industrial. Yeah. So in here is when we've analyzed the DNA. So this is a eukaryote, this is a bacteria, and archaea quite a separate group from two. Next one is also very important one in the book is eukaryote. U is true nucleus. So I just show the table showing the difference between prokaryote and eukaryote, and based on this you can see is some different characteristics of eukaryote compared with the prokaryote. So we have the nucleus, of course. This was the most important characteristic. And next one is they have the organelles, membrane organelles, like lysosome, like Golgi, like proteasome, and especially the mitochondria. And the size is also very bigger, a lot, 100 times. And they're younger, of course, because of the evolution from the simple one to the complex one. And about the DNA, we have a multi-chromosome in our cells, different with the bacteria. They have the circle plotting, and they only have only one, and together with some plasmid. And both we have a ribosome in there. Yeah. And next, this is a representative animal cells. You see that we have a very complex system. Like this is the most important. We have the nucleus in here, we have the Golgi, we have the ER, and anything else. We have the centriole, we have the mitochondria in here. Same for a plant cell, but a plant cell is very easy to recognize. We have the cell wall. So cell wall is very hard and make a plant cell very tough to break. So a plant cell, the cell wall plant cell like the backbone, like the bone of the cell, make the cell is more stable, and they protect the cells from the outside, right? But you may concern that animal cell is not, they don't have the cell wall why they still can survive, and they not breaking, because is the membrane of the cell is quite a very thin and very easy to break, but animal cell is not break yet. Very interesting, and you can see it's another chapter. We will have maybe chapter 11. We will discuss very deep about the cell membrane, and I also introduce a lot of things related with the application of that knowledge to biotechnology. How are the yeast cells? Yeast cells, you see in the table, they belong to the eukaryotic cells. And why? They look like a microbe. They're microbe because they're very small, but they not belong to the prokaryote. So the yeast cell is quite also, the organization is quite complex compared with the prokaryote. So they have their own mitochondria. They have the Golgi system. They have the nucleus inside, and of course they also have a chromosome, and they have a cell wall. So when I culture the cell before, I have some infection. I mean, some yeast cell come to my medium of animal cell culture. It's very hard to remove that kind of cells out of my medium. Okay. So yeast cell is a eukaryote, okay, not the prokaryote, but they still the microbe. So to finish the part of the classification, next we move to the organelles describe. So organelle describe is quite, you may be very familiar with this already from high school or from the first or second years of the university. So the organelle we have to divide two parts. So first is the two kind of the organelles. So first is derived from membrane, and the second is the bacterial-like organelle. What is bacterial-like organelles? So and what is the organelle of a cell? So that is a separate part of the cell inside of inside the bag or the box or something. They are kind of differ with another part of the cell. They specific, and they do specific function, like the cellular machinery. First, and the biggest one of the cell organelles is the membrane, the plasma membrane. The cells, whole cells membrane. Plasma membrane is differ from the membrane of the organelles. We have the organelle membrane also, but the same characteristic. We also the function of this plasma membrane is that they contain a cell. They contain a cell content. Without the cell membrane, the cell cannot be the cells, because you remember in the last when the one is looking and then the cells, and he named the cells under microscope is the cells, because he can see it's like the room next to the room, right? So the membrane is help to get the functioning correct. So the membrane that you know is that they have the layer of the phospholipid, and the most important is the protein in there. Phospholipid just provide you the separate the battery, but the protein is the and lipid also give you the some specific function, but a diverse function. You have to rely on a lot of type protein in there, a million type protein in the cell membrane. So the one information you need to know is you see a lipid quite a many, right? But the weight of protein in the cell membrane is the much heaviest part of the membrane. The heavy than, the sum of the protein weight in the membrane is the dominant in another part of the cell, like lipid or the carbohydrate. So using the electron microscope, human can take the picture of the membrane, and this is a 3D structure of the membrane. You have a lipid molecule, so by layer they have next slide will be better to explain this one. So this is the phospholipid. They have the polar characteristic. They have the head with the water loving, and the tail is by the fatty acids, and they don't like water. So they don't like water, and they face each other because they hide themselves from water. So they make the structure of the cell. And one of the very important function of the membrane is they control the movement of the chemical from the outside to get inside, from the inside to get outside. Without this kind of function, if the chemical can freely travel between out and in, so the cell is not different with outside, right? So more specifically, a few molecules move freely is water, carbon, CO2, oxygen, and ammonia. Actually, water is not moving a lot into the cells, but they can move in. Oxygen, of course, they can move in a lot, and CO2. And most of the chemical can go in and go out of the cell. Need to be go by the pore. The pore is made by the protein. We call it transporter or the channel. So because we have the next quick chapter, we will discuss about this one in very detail. So protein, they localize in a lipid bilayer, and they make the hole, they make the channel for deliver chemical out, in and in out. Okay, like this. So you can see that water, ammonium, oxygen, quite very easy to move out and in, free. Carbohydrate like lipid, they can go because of they can join to the layer of the lipid in here. So this molecules they also don't like water, so they can hide themselves in here, and sometimes they can transfer from the out to inside. And the glucose is not allowed to get in using the lipid bilayer route. They have to go to the by specific route by the transporter using some specific protein in there. And somehow they need to pay money to get in. Money is what? The ATP. And the ion like potassium ion or sodium ion or like this. Very important ion is proton. They completely inhibit, cannot move in. They have to move in using some channel or transporter. Okay, so That's all what you need. Information. This is 3D structure again. And here in the membrane we have like you see the protein, and this is some carbohydrate inside the protein. Some question like is how the protein can enter the membrane. Also, we can answer the question in the further chapter. And also, what is function of the cholesterol in here? Cholesterol also have important function of the cells. Not too much, but less is not good for the cells. We need cholesterol, but don't need a lot. Don't use a lot. Okay. Okay. Next is glycoprotein. Like is when the glycoprotein is a sugar, poly carbohydrate chain to add into protein in the post-translation process. After the protein made by ribosome finish, and some modification need to add into the protein. And very important part of the membrane protein is the enzyme. Some protein in the membrane, they have activity to catalyze some reaction. Next, we go to the cell wall. It's very detailed about the cell wall, right? Like we have the plant, we have the cell wall, we have the fungi, we have cell wall, and many protists we also have a cell wall. To function is protect the cells, but they have another function, like they can keep the cells is not growing so much, because if the cell grows so much, they will break. Like you see it, the chemical inside of the cells is kind of more dense. The concentration of chemical inside of cell is you have to admit that is a high concentration compared with the outside. So the water from the outside tend to be get into the chemical from the outside they tend to be get inside of the cells. So if they get inside more and more, the cell will be break. So the cell wall is try to resist with that situation, because they provide the pressure and the water cannot come in anymore. And that also happen in the animal cells, because animal cells are the same thing. The concentration of chemical inside of animal cells is also very high. So but the animal cell don't have a cell wall, so how can they manage that kind of thing? So the difference between the cell wall in the plant and fungi have chitin and the cellular. And somebody will also want to ask the question, like how cell wall can synthesize outside of the plant cell? Very complex system, very complex process. And next is a very important one is cytoplasm. So cytoplasm is all of the stuff inside of the membrane, but except the nucleus. So they can contain the component of the cells, that means organelles and another fluid, like the chemical inside of the cell. But it's very complicated in there. So like you see in the figure, you see the is the nucleus. You have the Golgi system. No, this is ER, and this is mitochondria, and this is a skeleton of the cells to make the cell is harder and more struggle. And but how about the space in here and here? A lot of space outside, different with the above description. In there is a water or something? No. they have a lot of chemical. This figure show like this one. They have a lot of stuff with the organelles and ground by various molecules, not water. Okay. Okay. Move to the next slide. So this is cytoskeleton. We have the one chapter talking about cytoskeleton. We have the three types of fiber to make the cytoskeleton, like the bone of the cells. Very complex. They we will come back this one later. Microfilament, microtubule, or intermediate filament. So this is a structure. So you see it like the cement. To build a house, we need the cement, right? So this is the same thing, like the iron in the building. So they have three functions. So mechanical support. So without the bone, without the cytoskeleton, cyto is the cell. So without the support from the cytoskeleton, the cell will be easy to break. That's why you see that the animal cell, even they only have the bilayer lipid, but they still very rigid. I mean, it's strong, because underneath of the cell membrane, we have a lot of the bone in there, and they keep the cell very stable. And another one is I call the organelles. So the organelles is they cannot moving freely in the inside of the cell. They cannot easy to moving because they fix the location by the cytoskeleton. So without some need, they not moving and they fix in place. And another one is that they have to move the substance. You see that the cell need to be take out some part and take in some part. So the back of the box or the back of the bacteria from outside or from inside, they move from this organelle to another organelles. They not moving by something very complex. Maybe you don't know about that before, but they need to move like the railroad of the train, like the train. The chemical moving by they move along with the string. They move along with the string. The string is made by the cytoskeleton. For example, when the rose ear, so the ribosome make the protein in there already, and that protein will be go into the Golgi system. The Golgi system be part in the box or the bag in the sac, and they move to the lysosome or move to the cell membrane to take out that kind of chemical. So they move on what? They move on the cytoskeleton. This is using some staining technology. You can see under microscope, a fluorescent microscope. We can see that the very complex organization of the cell cytoskeleton. It is a red, green, or the blue with different color. Any color is fine, but you see a lot of the string inside of the cells. This one is like a cilia and flagella is for help the cell can moving or take the food. Especially we need this for sperm, but they also make from the cytoskeleton, but in a specific organization. Next one is centrioles. Centrioles is also you know already. So they need centriole for separate two sets of the chromosome to the two daughter cells. But the question is, without centrioles, the cell can divide or not? And the answer is yes. Yeah. But some scientists try to remove centrioles and see the cells still can divide. And next we will talk briefly talk about some organelles of the cells, like to the nucleus. The nucleus is the most important organelles in the cell because they control the DNA. The DNA control all of the activity in the cells. In the bacteria, the nucleus don't have the bacteria. In the eukaryote, it's very complex. So the double membrane look like any organelles inside of the cells. They have a double membrane. Which one is single membrane in the cells? And they contain a chromosome. And next is a nucleolus. And you see it. In a nucleus, they have a nuclear pore. Nuclear pore is a very we will check the structure of the nuclear pore later, and they help the chemical can go in and go out of the cells, like some protein, and like I know is when the molecule go in or go out of the cell, we need to pay some money. I mean, some ATP can help the molecule can go across the nucleus membrane. Okay. This is our DNA. So from less condensed from the left side in the figure to the right side is the more condensed. Prepare for doubling. So DNA is a store all of genetic material in the cells, and we have a chromosome is DNA. Chromosome, not only DNA, we have the protein, a lot of protein, right? Remember histone, and they form for the cell division. And this is also very famous. I don't need to explain for that. DNA grow up around histone and highly organized, very condensed in the chromosome structure. And have a nucleolus. So somebody still maybe missing these organelles. Thus, most cells have two or more nucleolus. So they replace of the direct synthesis of RNA. So combine between the ribosome RNA and form the ribosome together with some protein to make the ribosome. With the main material is ribosome RNA, nucleolus. Next is ER, endoplasmic reticulum. So have to move some substance into the cells and network for interconnect with the membrane. So you see that ER is next to the nucleus. So some RNA can come out and then match with the ribosome in there, and they make a protein. And the protein will be go in back inside of the structure at the double membrane of the ER, and can transport to another organelles for using. See, right? Because remember the cell is a keep from when they divide, they're quite small, right? And they need to grow up. So grown up, they need to phospholipid, and they need the protein. They need the protein in the membrane, and also when the cells need organelle, need the ATP. So number of the mitochondria also increase. So where is the protein to make the mitochondria? Where is the membrane? I mean the phospholipid membrane. Where is it? What? So they synthesize inside of the endoplasmic reticulum, ER, rough or smooth. Rough is we have the ribosome in there to synthesize protein, and the smooth is not attack ribosome, but they have the enzyme to have the build molecule like a carbohydrate, lipid for cell membrane or the organelle membrane. Wait, we know the plasma membrane have a carbohydrate, but how about the insides, like the membrane of the organelles, like the Golgi system or another? We have the carbohydrate or not? That's also the question. And the Golgi apparatus is like a station for shipping and packaging. So the protein or lipid from the ER we move to Golgi, and from the Golgi they will attack and move some vesicle. In the vesicle we have a lot of chemical, and this will attack with some of the cytoskeleton and move to another destination like the cell membrane or lysosome or mitochondria, some another organelle. So go to the sac for storing the energy. Okay, so molecule come into vesicle, and vesicle fuse with Golgi membrane and modify the Golgi in there. So here is the some step of Golgi can extract, send out some chemical. This is ER. So after protein synthesize, protein is made from RNA making in the nucleus. Here, they come to the Golgi, and the Golgi will park and put some signal, and this signal will tell this packet will be go to where. They will go to the outside for exocytosis, or they go to another organelles based on the signal. And this is a lysosome. They contain a lot of digest enzymes, so they cut all of the material for the cells. So they have the cell renewer, because some protein, when we break the protein, we still have the amino acids. Or we break the lipid, we have the fatty acid. And also, even we can break down the cell organelles. And also the lysosome can help us to protect the cell by digest invader. So lysosome very important. So remember Nobel Prize in 2 years ago for the Japanese scientist. He discovered the autophagy. So this process is like the cells eat themselves, and then regenerate the material for another process of the cell need. So this is a very important process of the cells, and they relies on the activity of the lysosomes. And the vacuole. So the vacuole like the storage shack. So they store the water, they store the food, they store the waste, and waiting for take out. And more common in the plant than animals. Animal we have it or not? Yes, this here, adipocyte, the fat. This is the fat cells. We call it adipocyte. So here we have nucleus very small, and a very big part is and we have a fat inside of the sacks of fat. And like before that, I just think that my big belly is just have the fat freely moving in there. But now you see that any fat is storing its cells, and that cell is fat cells, and they big, and they contribute to my big belly. And adipocytes is a quite interesting target for a scientist can do the research, because of you see that anyone want to remove fat out of the cells, so the scientist need to identify the process, I mean the biochemical process or related to genetics, or how to make people remove fat out of the cell for the healthy. That's also very interesting. This is adipocyte. And now we go to the end about the bacteria like organelles. We talk about mitochondria and chloroplasts. So mitochondria is the place they make the energy by fire the molecules inside of the cells, like sugar, pyruvate, and chloroplasts is store energy. So mitochondria, they have their own DNA in the plasmid, about 15 kilobase, 15,000 base pair, around that I remember. So they bound by double membrane. They have the outside membrane, they have the inner membrane, and they have this interface between two membranes. And this is very important. We have a 2 chapter talking about mitochondria. So you see, is the mitochondria of the cell is I mean the number of the mitochondria is based on the need of the cell. If the cell need the mitochondria, they can make more mitochondria. They can synthesize it. And when the cell divide, different with the chromosome, they have to equally divide between 2 daughter cells. The mitochondria can divide quite also is equally, but it's not very strictly equal. And the mitochondria, we have talked to this history some billion years ago. So this is first we talk about the functional breakdown of the fuel molecules, and they release the energy in the ATP. And people and scientists talk about the possibility. The theory talk that before our ancestor is a non-use oxygen for living, we are pre-eukaryote cells, our ancestor cells. So they go place somewhere and they match the bacteria with using the oxygen for living. So they met each other and they engage. So they get into the cells, our ancestor cells, and they involve and they become the mitochondria. Very interesting, right? The bacteria living inside of other cells become an organelles of that cell, the essential organelles. So you see that before I'm talking about the phase of the genetic. So before the bacteria maybe is kind of they independently bacteria, they the living thing, they independently living. So they have all of the tool for living. They have many things to I mean the molecular tool they enough. But when they get inside of our ancestor and eukaryote cells, maybe a lot of that tool will be lost because they don't need anymore. They don't need to survive in the inside of in the environment. So they like sympathetic biology, symbiotic. So they benefit for each other. So our ancestor will be protect for the cells, for the mitochondria. So they don't need to some weapon to fighting with another environment. So that is symbiotic. And how about chloroplast? So the same thing. Interesting in evolution. So you see that in the early plant cell they have the mitochondria ready, so they go somewhere and they engage with another bacteria. Here is a photosynthesis bacteria, green bacteria. And they get enter, and they also become the another organelles for the plant. So the plant is giddy, right? So it's this is the kind of the rules of evolution. So it's the plant take the chloroplast later, before or after they got the mitochondria. So animal and fungi. And archaea is deep inside of the sea, so they don't need anything. They don't go any place for acquire themselves. And this is quite terrible, but the scientists say that this may be the very early ancestor. They don't have the mitochondria or something, or chloroplast. So we kind of finished the thing. So the question is, about the application. The scientists need to do the research about the cells. So the question is, can we extract or isolate the cell organelles or not? Because it's too small. The cell is always small, right? About 100 nanometer. But the organelle is too small. How the scientists can do the research using this thing? And the thing is, we can. So using the ultra-centrifugation. You know centrifuge, right? So first we need to break the cells, break the plasma membrane, but don't break so hard, because you can break the organelle too. And you put into the very high density molecules in the tube and use it apply for the very fast centrifugation. Guess what is the first component of the cell in the bottom first? That's the heaviest one. It should be the nucleus with the DNA. Remember, DNA is the most long and big molecule in the side of cells. And next one, we have the mitochondria. Mitochondria also very big. Another one is we have the also segment of the cell, like cell membrane or something. And what is the last one? Last one is should be the ribosome, quite a small organelles without the membrane. We have some organelles without a membrane inside of the cells, right? Ribosome. Okay, so we go a little bit more about chemical component of the cells. This is quite a difference between the earth and the cells. So you see that the cell very favorable for hydrogen, carbon, oxygen, and nitrogen. We have the 4 dominant atoms in our cells, in human body. And another ion is quite a very less, but essential. We need it. And to understand better about some process of the cell, is most of the molecular cell biology is we talk about molecules, and molecules need to do the reaction, right, inside of the cells. And any minute of the cell, we have a lot of type of reaction. So I'm a little bit touched with the chemical bond, like the most strongest bond in the chemical is a covalent bond, and the next is an ionic bond, and next is we have the hydrogen bond, and with the van der Waals or something, van der Waals. So all of the chemical reaction, we can talk about redox reaction, like reduction and oxidation reaction. So both they have one thing, the main thing. I'm just have a little bit briefly talk about biochemistry. So both all the process of the reaction, I mean the form, I mean making the link, making the bond. We need making the bond or break the bond of the chemical reaction. We need the electron moving. They jumping from this place to another place. Maybe they form or they break the bond. So in here is a covalent bond is the electron can move inside of the same same what same cloud. No, not same cloud. They share electron with each other, and in the ionic bond, so two electrons can move in one side, and another side is because the last electron, so they have the opposite charge. So what's make the electron moving? What make the electron moving? So we come to this table in the left side. You see, you remember something related with chemical. You know, any atom is we have some layer of electron, So any atom try to they tend to be get enough number of electron to each layer, and they satisfy and they don't have the reaction. So for example, argon in here, so they have the first layer have 2, second layer have A, the third layer have A. They fix already, so they don't need any more. So they don't join to any reaction, and they become the inert gas. And for example, like hydrogen is the only one, and they have to get loss of this one or gain more to get the 2 enough for the first layer. And C, very interesting, is carbon. Carbon have 4 in the layer number 2, and they can give 4 to another more atom to become the to get enough full of the first one, or they can get more 4 more electrons to be fulfill the second layer. So that is so they can join a lot of reaction, right? They can get loss or get gain of the electron. So that you can see that why carbon is a very active, and nature use carbon like the most used atom for a living thing. You see, it's carbohydrate. All of our cell material is from the carbon. So another molecule, another atom also very active for a living thing, like the phosphor, P. So they have 5, and they need 3 more. So they're very easy to join the reaction, like what? Like ATP, right? ATP is the molecules is they give you the energy. They're easy to break and easy to form to get you the energy. And this is maybe it's easy to understand with you. So you know is covalent bond is the most strongest one. And next one is we have non-covalent is ionic bond. And next one, the last one is not the last. We have the hydrogen bond. But have a look at here. So covalent bond is in the vacuum one without water, or in the water they don't change the pores. The link is very tight and strong. But ionic bond in the vacuum is strong, but in water they decrease a lot. And why is that? Somebody can help to answer that one. Okay, next one we talk about a protein. So now we have 2 protein, even though they have a structure is very fit each other, like the lock and the key. But the main reason to get these 2 protein get gathered together because of the charge. You see that the charge is any spot is kind of small, very small, compact. But when we combine with a lot of spot along with the structure of the protein, so the sum of the pork chart is very large, and they make the two proteins become each other and hard to break. That's kind of link. This is a small ionic molecules in the cells. So we have the sugar, this is a monomer for the polysaccharide, glycogen, and starch in the plant. While fatty acids, or this is a monomer, no, not monomer. That is also polymer, but we can make it more complex form in fat or some membrane lipid. And some of you remember the structure of the fat for the energy storing and the membrane lipid. And amino acids for the protein, like the polymer or nucleotide, making a nucleic acid like RNA and DNA. Very basic, right? So the how are the composition of the cell? So 70% of bacteria is water, and 30% remaining is we have RNA, protein, some polysaccharide like macromolecules. And this is just a test if you see the structure, the chemical is which is which. Like this one is a fatty acid, and they don't like water in this side. And this is a protein, polypeptide. This is amino acids. We have the N terminal, and this is C terminal. And this is a polysaccharide. You see it's a lot of glucose molecules in here, right? And in here we have the ADCG. This is should be the nucleic acids. So the bone in the living thing is a non-covalent bone, like the one-way or hydrogen, or a little bit ionic. And this is a anabolism process, right? Anabolism, not a catabolism. So ana is just make the polymer from the monomer. Monomer in here is a more complex form in here. In this process, we need energy to make this. Okay, next week we will talk about energy. I'll talk a little bit about chemistry. Okay, thank you very much.